Welcome back, dear students. Today our topic is anatomy of monocot stem. If you will take a transverse section of monocot stem and place it under the microscope, you will observe the following internal structural details. Starting from the periphery towards the center, the first region that you will encounter is epidermis. Epidermis is the outermost layer of cells, which is usually single layered. The individual cells of this layer are thin-walled parenchymatous. They are green, and on their outer side there is a thick layer of cuticle. And this epidermis also bears a few stomata at some places. This is very much important. In case of monocot stems, the epidermis bears stomata at some places, while as in case of dicot stems, there is no stomata in their epidermis. Then inner to epidermis there is one more region which is known as hypodermis. This is a multi-layered region which lies just below the epidermis, and it is made up of thick-walled sclerenchymatous cells. The main function of hypodermis is it gives mechanical support to the plant. But in some species like asparagus and wheat, the hypodermis is absent. Then the ground tissue. The whole tissue system which lies inside the hypodermis, except the vascular bundles, it constitutes the ground tissue. Okay, or you can simply say the whole tissues which lie inside the hypodermis, they form the ground tissue. And in case of monocot stems, the ground tissue is uniform throughout. It is not differentiated into cortex, endodermis, pericycle, and pith as is found in case of dicots. In case of monocots, the whole tissue system which lies inside the hypodermis, it is uniform throughout and forms the ground tissue. The individual cells of ground tissue, they are parenchymatous and possess distinct intercellular space. Then the vascular bundles. The vascular bundles in case of monocots, they are variable in size and number. For example, those vascular bundles which lie towards the periphery, they are small in size as compared to the vascular bundles which lie towards the center of the stem. And each vascular bundle is conjoint, which means xylem and phloem are present in the same bundle. They are collateral. Xylem is towards inner side and phloem is towards outer side. They are closed, which means they do not possess a layer of cambium between xylem and phloem, and hence we say they are closed for secondary growth. That means in case of monocot stems, secondary growth cannot take place because they do not possess a layer of cambium between xylem and phloem in their vascular bundles. And they are also endorch. Endorch means protoxylem is towards center and metaxylem towards periphery. And most importantly, all these vascular bundles, they lie scattered in the ground tissue. They do not form a ring-like structure as is found in case of dicots. In case of monocots, the vascular bundles are scattered. They do not form a ring-like structure. You have to remember this. And each vascular bundle, it is surrounded by a sheath of sclerenchymatous cells, which is known as bundle sheath. Here you can see in the image, this is the image which reveals some of the important anatomical features of monocot stem. The outermost layer is epidermis, then there is hypodermis, and the whole tissue which lies below the hypodermis, this is uniform throughout, and this is known as ground tissue. And inside the ground tissue you will find a large number of vascular bundles. They lie scattered, and the smaller vascular bundles, they are towards the periphery, and the large vascular bundles, they are towards the center of the stem. And if you will take a small section of this stem and see it here, you will observe some of the minute details. Okay, the outermost layer, which is known as epidermis, the epidermis is cuticularized on the outer side, and at some places you will observe some stomata there. Okay, but one important feature about the stems of monocots is the epidermis of their stem, it does not show trichomes or hairs. Hairs or trichomes are absent in case of monocot stems. Then inner to epidermis there is a broad region which is known as hypodermis, and in this hypodermis the individual cells are scleral chromatous, they are thick walled, but at some places you will find a large a few patches, okay, and these patches are chloran chromatous. And inner to this hypodermis the whole region is known as ground tissue, which contains a large number of vascular bundles which lie scattered in this ground tissue, okay. The vascular bundles towards the center they are large in size as compared to the vascular bundles which lie towards the periphery. And each vascular bundle, as I have already told you, it is conjoined, collateral, closed, it is end arc, okay. And one very important feature about the xylem of this vascular bundle of monocots is it is Y or V shaped. It is Y or V shaped, in which the two extended arms they are represented by two vessels, and this region is represented by two more vessels. But the difference is that these two vessels are large in size as compared to these vessels which are smaller in size. Here you can see these are the large vessels, okay, and these are the small vessels. And one thing that you will remember about the xylem of monocots is the xylem parenchyma is absent, because the xylem parenchyma cells they get destroyed, okay, they get dissolved and result in the formation of a cavity which is known as lysogenous cavity, okay. So this is all about the anatomy of monocot stems. I hope you all understand this. Thank you and stay blessed.